Hi, everyone. Thank you, ADAA, for the opportunity to share our research. Um, we hope that it's going to be useful to you either in your teaching capacity, your research capacities, or in your clinical work. The origins of this work actually were many years ago when I had an administrative appointment. And as part of that appointment, I would talk to various constituencies around the state about the impact of the university on the state's economy. One group I talked to was a group of African American businessmen in rural Georgia. And they said to me, "When you're not doing this stuff, what do you do?" And I said, "Well, I'm a child psychologist. I work with children and families, and I do research on uh, parents and children." And they said, "What do you know about our families?" And I went through the Rolodex in my mind, and I didn't know very much about rural African American children. So when I finished that role and went back to my faculty role, the first thing I did, like most researchers, is I wrote a grant to NIH to study rural African American families. Um, Sent it off to NIH. It was immediately trashed. Felt badly for a while. Took the proposal, put it in an envelope, and sent it to the Spencer Foundation. And people at the Spencer Foundation liked it. They funded the work. And then that led to future funding from NIH, which has continued to this day. And the original work we did with rural African American families mapped the developmental trajectories of rural African American children, adolescents, and young adults, and identified the risk and protective factors that were responsible for variations in these trajectories. And we took the protective factors that we found in our research and used them as the ingredients for prevention programs for African American pre adolescents, adolescents, and young adults. So, in our time together today, we're going to talk about the rural South as a developmental context for African Americans. We're going to talk about testing the weathering hypotheses. What are some explanations for the development of health disparities in young people? And finally, we're going to talk about the health benefits of family centered prevention programs. This is the southeastern Black Belt, with stretches from North Carolina, South Carolina, Georgia, Alabama, Mississippi, Louisiana, touches Oklahoma, and a little bit of Texas. 35% of the nation's poor live in this area. More than half of rural African American children grow up in poverty, who grow up in the Black Belt. Um, the educational systems are underfunded. Access to health care is limited. There's not a lot of dentists in these places. And it's a challenging place to raise children. So, in our work with African American families, we decided about 10 years ago to start focusing on health outcomes and not just mental health outcomes. Because so many of the caregivers of the children we were studying in our longitudinal samples were developed chronic diseases, symptoms of heart disease, type 2 diabetes, high blood pressure. Um, Other relatives had strokes. And we wanted to know, what is it that goes on in African American communities that both protects young people from the development of early signs of later illness, and what are the risk factors? And those are detailed in what's called the weathering hypothesis. In these studies that I'm going to describe, there are three outcomes you're going to hear about. One is allostatic load, and it's a marker of chronic physiologic stress and wear and tear in the body. The way we measure it is we get overnight urine voids from which we assay cortisol, epinephrine, norepinephrine. We draw some blood to get markers of inflammation. We take blood pressure, we get body mass index, and we combine these into a cumulative index called allostatic load, which forecasts cardiovascular disease, diabetes, stroke, and earlier mortality. We also um, get data on genome-wide methylation, from which we obtain a marker called epigenetic aging. And the difference between your epigenetic age and your chronological age is a measure of how accelerated aging is at the cellular level. Um, this turns out to be a remarkably predictive uh, uh, variable for later health outcomes that include cardiovascular disease, diabetes, cancer, and all causes of mortality. We measure metabolic syndrome, which is something that your physician probably measures when you go get your yearly physical. It's a cluster of risk factors driven by abdominal adiposity, includes high blood pressure, impaired glucose control, and lipid dysregulation, and it predicts a whole range of cardiometabolic diseases. So the Great Recession took place between June 2007 and December 2009. And as it turned out, one of our samples of approximately 400 to 500 people, depending upon where we uh, analyzed data, lived through the Great Recession. And so they were 16 at the beginning of the Great Recession and 19 at the end of the Great Recession. And so we looked at to see if changes in economic hardship were associated with biomarkers of health. And we formed three groups: a stable low hardship group. They never dipped into poverty across the Great Recession. A downward mobility group who started out not living in poverty, but slipped into poverty during the Great Recession, and a stable high hardship group who lived in poverty across the entire Great Recession. And what we found when we looked at the biomarkers I just described for you is cardiometabolic risk is really allostatic load. And we found, not surprisingly, those young people who lived in poverty across the Great Recession showed the highest levels of allostatic load, followed by those young people whose families slipped into poverty across the Great Recession. And the lowest levels were people who never lived in poverty whatsoever. The same patterns were found for two different measures of accelerated cellular aging. Those young people who at age 19 and 20 lived in poverty across the Great Recession uh, evinced the highest levels of accelerated cellular aging, followed by children from families who slipped into poverty, followed by families who never lived in poverty across the Great Recession. So these data show, and it's a way of showing via natural experiment, how changes in economic hardship are associated with different biomarkers that forecast health. And interestingly, when we look at the self reports of overall health status, those young people who grew up in the stable low hardship group and the downward mobility group said, I feel the same, when in fact their biology looked very different. And those young people who lived in poverty across the Great Recession showed the highest levels of health problems. We followed these people up in 5 years, when they were 25, looked at the same groups and looked at their metabolic syndrome. And the findings paralleled those I just described, we found 5 years earlier. Again, those young people who lived in poverty across the Great Recession had the highest levels of metabolic syndrome, followed by those people who slipped into poverty across ages 16 to 19, and followed by people who were never in poverty at all. And again, this is another demonstration using a natural experiment of how economic hardships in the families that children grow up in are reflected in their um, health under the skin. One hypothesis that hasn't received very much attention is how race-related stressors impact or forecast during uh, adolescence uh, physiologic dysregulation. And so we did that in this study, and the way we did this study is across ages 16 and 19. We had about 400 uh, participants who, across those ages, uh, gave us information about how frequent their encounters were with racial discrimination. And their parents, across that same time period, also provided us with information about how much supportive caregiving the children received. And what the data show is that those young people who experienced high and stable levels of racial discrimination and didn't have the benefit of supportive caregiving across that time showed the highest levels of allostatic load at age uh, 19. Now, so these findings support what are called the parent effect hypothesis found in the animal literature, in which positive caregiving tendencies forecast um, uh, facets of brain functioning, neurodevelopment or neuroendocrine functioning, and immune function. One hypothesis that also has gone untested in the health disparities literature is that exposure to racial discrimination will prematurely age cells and tissues, resulting in lower life expectancies. And we tested that hypothesis, and again, the parent effects hypothesis from the animal literature. And to do that, we obtained measures of accelerated cellular aging. Um, we also, once again, obtained data on supportive family environments. So for this first sample, they were 16 to 18 when we obtained data on their exposure to racial discrimination. And we again formed two groups uh, via some using some statistical techniques: those who were exposed to high and stable discrimination, and those to low and increasing levels of discrimination. And we looked at the supportiveness of their family environments. And what we found is those young people who were exposed to high and stable racial discrimination um, showed accelerated cellular aging, but not when they had receipt of supportive caregiving. Now, we replicated this in another sample, and what you can see is the distributions look exactly the same. The differences were this sample were 17 years old at their first assessment and 19 at the second assessment of racial discrimination. So we looked at the trajectories of racial discrimination across ages 17, 18, and 19 for this sample. And at age 22, we obtained a blood draw from which we assayed the degree of accelerated cellular aging. And once again, we obtained across ages 17 and 19, assessments of the, their receipt of supportive caregiving. And the findings are remarkable. Um, they show the highest levels of accelerated cellular aging were among those young people um, who were exposed to high and stable levels of racial discrimination, and they didn't have the benefit of supportive caregiving. We also, at the Center for Family Research, developed three prevention programs, which I'll tell you about in a second. And we wanted to see if they had any kind of health effects. They were developed to deter drug use and abuse and engagement in risky behaviors. And they're very efficacious to do that. But we wanted to take the next step and see, are there some health benefits for these? So we developed three different family centered preventative interventions. The first one we developed is a Strong African American Families Program, which was developed for pre adolescents age 11. Second program is a Strong African American Families Teen Program, which was developed for 15-year-olds, for young people as they entered high school. And the last program, Adults in the Making, was developed for high school seniors to arm them with some protection before they go off and leave high school and often into a world that doesn't have a lot of educational and occupational opportunities. Each of these programs have been tested in randomized clinical trials. They are efficacious for at least 2 years in deterring the development of drug use and the escalation of drug use. We have followed the safe people from the time they were 11 through age 25 and have built in different measures in these clinical trials that I'm now going to share with you. So this is an analysis we just finished, and it's going to appear soon in print. And we asked a pretty simple question. That is, if at baseline in the Strong African American Teen Program and at baseline in the Adults in the Making program, young people encountered high levels of racial discrimination, could participation in those prevention programs deter the development of mental health problems? So take a look at panels A and B, and what they show are the increases in conduct problems for young people who experienced high and low levels of racial discrimination at baseline, who were or were not assigned randomly to a prevention condition. And importantly, it shows their increases in conduct problems. And across both prevention trials, the data show that those young people who were exposed to high levels of racial discrimination and were assigned uh, to one of the prevention programs, they did not increase their levels of conduct problems across 2 years, whereas similar young people in the control conditions did. In this panel of findings, we looked at depressive symptoms and symptoms of anxiety, and we found that for participants in the Adults in the Making program, those young people who were assigned, uh, who had experienced high levels of racial discrimination at baseline and were assigned randomly um, to receive the prevention program, evinced lower levels of increases in depression and anxiety than in similar young people who were in the control condition. So here we show that participation in family-centered prevention programs across two programs show fairly similar results in deterring the development of conduct problems, and in the case of AIM, depressive symptoms and anxiety symptoms. And not shown here were some other analyses that indicate that increases in supportive caregiving by each prevention program were partially responsible for the protective effects. This was our first attempt to look at the beneficial health effects of participation in one of our prevention programs. Um, Pro inflammatory cytokines are implicated in all of the cardiometabolic diseases of aging and are also influenced by exposure to stress. So we thought Um, at age 19, we would draw some blood. We would assay pro inflammatory cytokines. We assayed 6 of them, you can see on the right side of the slide. And then we combine them on the left side of the slide as a composite. And we were able to show that participation in SAFE at age 11 deterred levels of pro inflammatory cytokines for those young people who participated in SAFE. And here again, we did mediational analyses which showed that increases in supportive caregiving sponsored by participation in SAFE were partially responsible for the prevention effects. In this paper, we focused on accelerated cellular aging and whether young people grew up in families where during pre-adolescence their mothers reported elevated levels of depressive symptomology, and wondered if growing up in a household where the parent reported elevated levels of depressive symptomology at age 11 would forecast increases in cellular aging that were detectable at age 20. And we found that to be true. Then we asked, would this occur for young people whose families participated in SAFE? And we found that it did not. That young people who grew up in households where the parents evinced high levels of depressive symptomology and participated in SAFE did not show high levels of accelerated cellular aging. And finally, we asked, um, was this mediated by SAFE's decreases in the levels of harsh parenting that the young people received? And that's exactly what we found. It's not unusual that people who are feeling badly try to limit interactions with their children. And one way they do that is to engage or use uh, harsh disciplinary practices. And we found out that participants in SAFE decreased their parents in SAFE decreased their use of harsh disciplinary practices, and that was responsible for the protective effects of SAFE on deterring higher levels of epigenetic aging. I think this is an important finding. We took a little bit different tact in this one. I wanted to focus on prediabetes because prediabetes is so very prevalent in rural African American communities. And there's literature to suggest that um, when children have more adverse childhood experiences, they show higher levels of the stress hormones cortisol, epinephrine, and norepinephrine, and greater concentrations throughout the body of those stress hormones influence glucose metabolism. and glucose metabolism is a is responsible for pre-diabetic status. So I wanted to see if young people who experienced more adverse childhood events and were assigned randomly 14 years earlier to the SAFE prevention program would not show the same levels as similar children who experienced frequent or more adverse childhood experiences in the control condition. And you can see from looking at the figure that actually that did turn out to be the case. Very strong protective effects of experiencing SAFE 14 years earlier. Finally, I want to show you a set of slides which asks the question, A, does living more years in poverty across adolescence, is that associated with neurodevelopment as defined by uh, lower volumes of the amygdala and areas of the hippocampus? And B, could that association be ameliorated by participation 14 years earlier in SAFE? And here you can see that young people who spent more years in poverty and were in the control condition showed, with increasing levels of time spent in poverty, smaller amygdala volumes. And the same thing was found for the hippocampus. But those young people who experienced more years in poverty and participated in the SAFE intervention showed no effect of poverty on their neurodevelopment. And here are the findings for hippocampal areas. So in this brief presentation, I wanted to show you different ways in which our prevention programs can deter uh, different events, stress from getting under the skin and into the brain. So today, so far, we've linked contextual challenges to allostatic load, accelerated epigenetic aging, and metabolic syndrome among rural African Americans. We illustrated the buffering effects of supportive caregiving. We demonstrated that implementing programs that enhance resilience through strengthening family relationships may play an important role in improving public health. Couldn't have done this work without the support and confidence of these NIH institutes. We are very grateful. Likewise, science is a social process. It takes a village, and I have wonderful colleagues. I'm grateful to them as well. I just wanted to show you we are proud that we received from the Society for Prevention Research the Advances in Culture and Diversity and Prevention Science Award. And notice that it was for the Family Research Center team, not any one individual. Now, the most important slide in the deck. These are all the people at the Center for Family Research who made what you see possible. None of this can be done. My colleagues here form relationships with communities across the rural parts of Georgia. They develop trusting relationships with thousands of families. They collect data that is not easy to collect. They organize the data. They analyze the data. And I am truly grateful to them. And on behalf of all of them and myself, thanks for letting you share our work with you today. Talk to you soon.